So now that we've got our manolo-CoA, which has the extra carbon attached at the end here, with the addition of the carbon dioxide to give us the extra carboxylic acid group, we then essentially attach the ACP in the same way that we attached it to the acetyl-CoA. So it's just a matter of removing the CoA and attaching the ACP, which is our acetyl carrier protein, in its place. So we can just think of the acetyl carrier protein as a protein that will be able to attach to our acetyl group. And this time, this is what I've drawn here, is the acetyl ACP. So we actually have an extra carbon that is attaching as well in our manolo ACP. So we've still got that carbon that's double bonded to oxygen. This time we've got two hydrogens attached to the next one, and then we've got that carboxylic acid group at the end here. So this is our manolo ACP. So to create the manolo ACP, essentially we just remove the CoA and attach the ACP in its place. So that's what we can see on the right hand side here. We have the manolo ACP and the CoA has now been released. So essentially, to start the lipogenesis process, we need to do two things. We need to generate the manolo group by adding the carbon dioxide group to an acetyl-CoA, and then we need to take the CoA off and attach it to ACP. And we need to do that step with another acetyl-CoA to create the acetyl ACP group. So now we've got our two starting points that I've drawn here. We've got the acetyl ACP and the manolo ACP. Now we can actually begin the lipogenesis reaction. So basically this is the stage that we call chain elongation. So it's four reactions that can be used to convert our ketone group, C double bond O, into a carbon that just has hydrogens attached. So essentially we're removing our carbonyl group and turning it into a carbon that has just hydrogens. So remember, our structure of the fatty acid just contains the carbons and hydrogens attached. So there's no other oxygens or OHs except for right at the end where we have that carboxylic acid group. So essentially what we're doing is we're removing these carbonyl groups and turning them into the CH2s. So to do this, we use an enzyme called fatty acid synthase, and the end result of this process is the production of a fatty acid. So we have to do lots of cycles of the chain elongation. So every time we do one cycle of this lipogenesis, we're actually adding two carbons. So each cycle will grow the fatty acid by two carbons. And then once we've got that fatty acid that is two carbons longer, we can add it back into another lipogenesis cycle to extend it by another two carbons. So it will make sense how this works when we look at the individual steps. Alright, so now that we have these two starting compounds, the manolo ACP and the acetyl ACP, then we can do the first step of the chain elongation phase of lipolysis, where we combine the acetyl ACP and malonyl ACP to create what we call aceto-acetyl ACP. So we call this a condensation reaction, which basically just means we're bringing them together. So that we're attaching these together. So we'll actually have one of the ACP proteins being lost because we only have a single ACP protein in the product here. So you'll notice that the acetyl ACP is a 2-carbon compound, manol is 3, and the acetyl acyl ACP is 4 carbons. So we've lost a couple of carbons, and we'll see exactly how this reaction works in the coming slides. And then after we've created the acetoacyl ACP, then we do the steps 2 to 4, which are basically the reverse of the functional group changes that we see in beta oxidation. So we start off with our ketone group, which is a carbon that is double bonded oxygen, the carbonyl, and then we're attached to 2 other carbons. So we do a hydrogenation reaction via reduction, which is meaning we have a redox reaction, and that is facilitated by our NADPH. So now, instead of being a ketone group, it's a secondary alcohol, where we've got that OH bearing carbon attached to 2 other carbons. We then do a dehydration reaction. So this is where we will be removing our water. And by removing water, you turn the alcohol into the alkene. So by taking off the OH and the hydrogen that's attached to it, the adjacent carbon, we use that to create our water, and that means we have less atoms to form bonds with, so the carbons will form an extra bond together, and that's what we see in the alkene part of the structure here. And then the last step is another redox reaction, where again we are doing a hydrogenation, so adding our hydrogen to remove that double bond, and in place of the double bond attach it to an extra hydrogen. So we'll have a look at each of these steps individually. So step 1 is the condensation or combination of the acetyl ACP and the manol ACP. And we're getting those to combine to create the acetoacyl ACP, which it contains those 4 carbons. So notice that it's not just simply the addition of the 5 carbons, because we've got 2 carbons in the acetyl ACP and 3 in the manol ACP, but the acetoacyl ACP actually only has 4 carbons. So 1 carbon is actually lost as carbon dioxide in this process where we do the condensation reaction, and we also jettison or lose one of the ACP groups. So the carboxyl group that we had added to create the manol ACP, shown here in purple, is actually lost as carbon dioxide. So as we have that component being lost, the CH2 can actually form a new bond with another component. And what it forms the new bond with is actually the acetyl group that is attached to the ACP unit. So we have the ACP unit from the acetyl ACP being lost, and then we can get the fusion of these 2 groups in blue here. So we have that acetyl group, which is here, and this acetyl group was originally a part of the acetyl ACP. So originally from the acetyl ACP, and then we've got the rest of the structure, which was originally from our manol ACP, and we can connect because there's been the loss of that carboxylic acid group, which gives us that new position to form the bond. So we've got that new bond forming between the acetyl group and what remains of the manol ACP. So together, the 2 carbons of the acetyl group and the 2 carbons left of the manol ACP combine to form our aceto-acetyl ACP. And then this is the group, the aceto-acetyl ACP, that we actually use to generate our fatty acid, or at least our first 2 carbons that have single bonds between them. So this is what we will input into those functional group change reactions. So what is actually happening in this first reaction of the lipogenesis cycle? So essentially, the acetyl ACP and the manol ACP are coming together to form a new compound called aceto-acetyl ACP. So the aceto-acetyl ACP contains 4 carbons, and to create this compound, we've lost the carboxyl group, which was a part of the manol ACP, and that's been lost as CO2, and the ACP has also been lost from the aceto-ACP. So the loss of these groups basically allows us to attach the remaining carbons from the manol ACP and the acetyl ACP, and that's what we have formed in the aceto-acetyl ACP. So now that we've got this aceto-acetyl ACP, we can actually start modifying the group that we show here in the orange color. So this is our ketone group that is going to be modified to our alcohol. So that's what we're doing in this reaction. We're doing a hydrogenation reaction, which is the addition of hydrogen, and that's allowing us to convert our ketone group to our secondary alcohol. So whenever we see one of these coenzymes, we know that it has to be a redox reaction. So we haven't seen this type of coenzyme before in any of our other metabolite reactions, but essentially it's very similar to NADH, but it just has that extra phosphate in there as well. So basically what happens is we're going to have the coenzyme being oxidized in the redox reaction, and we'll have the aceto-ACP, specifically the region here shown in brown. This is going to be reduced. So the reduction of the ketone is facilitated by the oxidation of this coenzyme. So essentially what happens is we take the hydrogens off the coenzyme and put them onto the carbon here. So we can see where those extra hydrogens have been put. So we've got one associating with the oxygen and another one associating with the carbon. So now we don't have our carbonyl group, which is a part of the ketone. We've now got our OH group, which is our alcohol. So what makes the group we've created here, the beta-hydroxybutyryl ACP, what makes this a secondary alcohol? So remember, to work out whether something is a primary, secondary, or tertiary alcohol, you look at the carbon that is attached directly to the OH group. So that is this carbon in the center here that we've just modified. And then from there, you look how many other carbons that carbon is attached to. So we have 1 carbon on each side. So overall, there's 2 carbons. So this makes this a secondary alcohol. So now that we've created our beta-hydroxybutyryl ACP, we can undergo our third step of the lipogenesis cycle. And this is where we perform a dehydration reaction. So remember, dehydration means removal of H2O. So we need to find within our beta-hydroxybutyryl ACP the atoms that make up water, because we're going to remove them. That's what the reaction says. It's a dehydration. So we're going to actually lose this OH group that is a part of our compound here, and we're also going to lose one of the hydrogens from this carbon that's next to it. So if we take off that OH and another hydrogen from this carbon, what do we have? What have we made if we combine OH and H together? We now have made H2O. So we've taken away the atoms that make up our H2O compound. So that gives us this arrangement where the carbon each now only has the bond to one hydrogen and also the other carbon in the chain. So each of these carbons that I'm putting the boxes around that we've just modified only contains 3 bonds. And remember, all carbons need to contain 4 bonds. So the answer to this is for the carbons to form a second bond between each other. Now it's forming a double bond. So what do we call a group that has a double bond? So we call this an alkene group. So we've turned our alcohol into our alkene. And we've done this by removing our H2O. So once we have turned the alcohol into the alkene, we now call this the cantonal ACP. So don't worry too much about the individual names for these compounds where we're modifying the functional group. The important thing is to know the modifications of the functional groups. So we start with the ketone, and then we convert the ketone to the secondary alcohol, and we do that via a redox reaction where we are reducing the ketone. And then the next step that we just saw here was our dehydration, and that gives us an alkene. So the interchange between the functional groups is the important part of these steps, not the actual names of the intermediates. And then once we've got our alkene, we're getting very close to creating our carbons that just contain carbons and hydrogens and no double bonds and no oxygens attached. So remember, that's what we need in our long chain fatty acid that contains all of these carbons and hydrogens, which are represented by these lines. So the last step is basically to remove this double bond, which is actually in the trans configuration, and we can do this by undergoing another redox reaction. So we know it's a redox reaction because we've got the coenzyme involved. And again, the coenzyme involved is our NADPH. So the hydrogens from the NADPH will be donated to the cantonal ACP where we have this double bond. So that means, just like our other reaction, we're getting oxidation, loss of hydrogen and electrons from our NADPH, and that means we're actually reducing the the section of our cantonal ACP, which is shown here in blue. So remember, all redox reactions contain one component being oxidized and one component being reduced. So the outcome of this reaction is what we see on the right hand side here. We've now got our butyryl ACP, which contains only single carbon-to-carbon bonds. So we've removed the double carbon-carbon bond that was here, and we've done that by adding hydrogens in its place. So basically, we've taken the double carbon-carbon bond and converted it to a single carbon-carbon bond by attaching hydrogens to those carbons instead. And remember, the source of those hydrogens was from our coenzyme NADPH. So now we've got what we call butyryl ACP. So if I drew this in the line diagram, remember each point on this line represents carbon. So we'd have carbon 1 going to carbon 2 and then carbon 3. and then we've got our C double bond O S ACP. So this is another way we can represent the butyryl ACP. So where we have these lines here, this is what we have generated in the lipogenesis reaction. So essentially we've generated carbons and hydrogens linked by single bonds. And that's what we need to form our fatty acids. Remember, our fatty acids contain these long chains that are just carbons and hydrogens. Some have double bonds. That's if they're unsaturated, but we're making a saturated fatty acid here. So there's not going to be any double bonds. And at the end of the fatty acid, we've got that carboxylic acid group. So essentially what we're doing in our lipogenesis process is making these carbons and hydrogens that are just linked by single carbon-carbon bonds. And the next cycle will actually add on another 2 of our carbons. So there'll be another carbon here, another carbon here. Doing the cycle again, we'll add another 2. So we're going to have a look at that in a little while. But essentially every time we do this cycle, we add another 2 carbons that contain only single bonds between them.